Good afternoon, or good morning, or good other time of the day, depending upon where you are. So on behalf of the Biochemical Journal, the Biochemical Society and Portland Press, and on this occasion, the Biochemical Journal, I'd like to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. And each of the webinars will give you the opportunity to ask questions via text, and we welcome suggestions too for future topics and speakers to feature in our webinar series. Please see the website you can see on the bottom left, society website for more details. And so my name is Mark Lemon. I'm a professor of pharmacology at Yale University, and I used to be chair of biochemistry and biophysics at UPenn before I moved to Yale. And I'm also the co-director of the Cancer Biology Institute at Yale. And my role with respect to the Society is to chair the editorial board of the Biochemical Journal. So a special welcome from me. Today's webinar is entitled Tackling SARS-CoV-2 Biochemistry, and it's run as part of Biology Week. This session is focused on the collection of 7 original papers that we published in the Biochemical Journal, where John Diffley and his colleagues, from whom you'll hear in a few minutes, present the findings for all seven actually of the non-structural SARS-CoV-2 enzymes, the enzymes that are responsible for replication of the virus. So I'd just like to say a few words about the Biochemical Journal. I took over as chair of the editorial board at the beginning of January this year, and I'd been associated with the journal for a long time. It's a journal with a terrific scientist group that runs it in terms of editorials and so forth. And our mission broadly is rigorous understanding of molecular mechanisms in biology, which is at the core of the Biochemical Journal. And as a journal, we play a key role in advancing understanding through publication of high impact research in 21st century biochemistry. And we're in the process at the moment of adding a variety of elements of 21st century biochemistry, including new systems biology section and so forth, and themed review issues. So look for some changes over the next little while. Many advantages to the journal. I actually just had a paper published there a couple of weeks ago, which was remarkably efficient compared with other papers that we're publishing elsewhere. Quick decision, free to publish using a standard license, no page fees, publication charges or color fees. And open access with no author-facing fees using the read and publish offerings that more and more institutions are taking. But the key thing that we try to advocate and do is painless publishing. Unlike other journals, we won't get you to do experiments that reviewers have a whim that you should do. So extra experiments not necessary to the paper will not be asked for, which actually makes publishing, you publish your paper. Rapid listing in Medline and etc. And it's an independent society-owned journal for your biochemical society. So next, let me introduce the subject of today's webinar, which is, as I said, tackling SARS-CoV-2 biochemistry. And the beginning of the COVID-19 pandemic launched a race across the world to produce effective vaccines and other therapeutics. Several of the vaccines have now been rolled out with remarkable speed and effectiveness. But we all know that frequent emergence of SARS-CoV-2 variants serve as an important warning that this is going to be an ongoing battle. The needs for vaccines will change. New generations of vaccines will be required, and what's also clear is that effective antiviral compounds are going to be crucial tools in the long-term armory against newly emerging virus variants. Now, to meet this challenge during the 2020 COVID-19 lockdown, and to take advantage and to make the most of that time, John Diffley and his colleagues, both at the Crick Institute in London and the University of Dundee, retooled their laboratories during that period to identify some new small molecule inhibitors of the enzymes that are required for replication of the SARS-CoV-2 virus. They focused on pre-existing pharmaceuticals in doing this, with the hope that the inhibitors that they find could be deployed rapidly. in response to COVID-19. And we just published a couple of months ago a collection of 7 original papers in the Biochemical Journal describing the findings for 7 of these SARS-CoV-2 non-structural enzymes. And this collection describes several drug candidates that will serve certainly as important starting points for further development and/or tool compounds for studies of the vagaries of this virus that's terrorizing us all at the moment. And so during this webinar, the speakers will describe the biochemical screens that they used to identify inhibitors of the SARS-CoV-2 encoded enzymes. John Diffley will begin by providing an overview of the project. Jennifer Milligan will then describe the screen for NSP5/M-Pro inhibitors. Kang-Wei Tan will then describe work leading to the NSP3-PL-Pro screen. Berta Canal will describe screens for NSP non-structural protein 14, 10, and 15. And Raquel Hilfer will then describe how she tested the inhibitors that were identified with those approaches in viral growth assays. So that's our list of speakers. Before I hand over to John to get the talks kicked off, I'd just like to mention a little bit of housekeeping in terms of submitting questions. And the questions will be asked at the end of the webinar, not between talks. But please do send your questions in at any time. So if you have a question, if you could type it in the question box as shown here on the figure, and then if you could specifically state for who your question's for, and we'll try to answer as many as we can at the end. They'll come through in a little box to me and Sandra, and then we can chair the asking and answering of the questions by the panelists. So I hope that makes sense. And your questions, I should point out, will only be visible to the organisers. So you won't be distributing across to everybody. Okay, good. So I think that's everything. I think we can now get started. And the first of our invited speakers today is Dr. John Diffley. John was born and raised in the New York City area, where I currently am actually today. He obtained his BA, MS, and PhD from NYU, New York University, and then worked as a postdoctoral fellow in Bruce Stillman's laboratory at Cold Spring Harbor from '84 to '90, where he started working on yeast DNA replication. John then moved to the UK to Clare Hall Laboratories Imperial Cancer Research Fund in 1990 as a junior group leader. Was promoted to senior group leader in '95 and became director of the Clare Hall Laboratories and deputy director of the London Research Institute for CRUK in 2006. Then with the founding of the Francis Crick Institute in 2015, John became associate research director, with responsible for junior researchers, students, postdocs, and etc. And in '98 honours, he was elected a member of the European Molecular Biology Organization. In 2005, he was elected as a fellow of the Royal Society, and in 2020 to the US National Academy of Sciences, so the National Academies of the UK and the US. John won the Paul Marks Prize for Cancer Research in 2003, the Louis Jeantet Prize for Medicine in 2016, and the Gardner International Award in Canada in 2019. So thank you, John, for doing this and for overseeing these papers. It's a great pleasure to have you speak and kick off tackling SARS-CoV-2 biochemistry. Thank you.
Great. Thank you very much, Mark. Can you all see and hear me?
Everything's good.
Everything's good. Great. Okay, well, I guess I first want to start by thanking the Biochemical Society for organizing this. I think it's a really great thing for all of us to kind of bring sort of an end to what's been a very challenging project. I also wanted to really thank the Biochemical Journal for publishing these papers, and I guess reiterate what Mark said. It's a great journal, and they didn't ask us to do things that weren't necessary to get the papers published. So thank you very much, Mark, and also Dario Alessi, who was involved in this as well. And then I'd also like to thank the Francis Crick Institute, because it was a lot of the facilities and personnel here that we used quite a lot during this period. So I just start with those few thanks. Okay, so I guess the COVID-19 pandemic is not a surprise to anybody. It's been an atrocious thing. Over 200 million cases, 4.5 million people have died from it. And at this point, back in March 2020, we decided, my lab and I decided that we really wanted to try and do something positive during this, so where we could actually use our expertise. And so we set out to try and identify some small molecule inhibitors of the enzymes that are encoded by SARS-CoV-2 as potential antiviral drugs. And really, we had a number of goals. We wanted to, a number of reasons for wanting to isolate small molecule inhibitors. One is to be able to exploit these chemicals ultimately as tool compounds to study the biology of the virus, to identify drugs potentially for repurposing, to identify lead compounds for further drug development, and also to identify synergies between inhibitors for some potentially novel therapeutic strategies. So I just wanted to start with this overview of the SARS-CoV-2 genome. I think you can, let me make my laser pointer here. I think you can see up here that the genome is about 30 kilobases, and the first, you probably know this spike protein here, S, is encoded by this last third of the genome. But what's really striking is this massive open reading frame, ORF1A, 1B, which really encodes two proteins, one 400 kilodalton protein from ORF1A, and then a 700 kilodalton protein from a program frame shift here that encodes ORF1A and 1B. And this massive protein gets cleaved by two proteases that are encoded within it, NSP3 and NSP5, into 16 what are called non-structural proteins. And these really have all of the enzymatic activities that are involved in replicating the virus. So there's a whole bunch of enzymes encoded. There are actually 9 of them. Two of the proteases I've already mentioned, NSP3, part of the NSP3 protein, and NSP5, and you'll hear from Kang Wei and Jennifer about the screens that they did. I just want to note that in addition to processing the polyprotein, the NSP3 protein also has isopeptidase activity against ubiquitin and ISG15, which is probably important in innate immunity, in avoiding innate immunity. There's a mono ADP ribosylase. I won't say much more about that. There are two cap methyl transferases. So these put methyl groups on the 5' cap of the messenger RNA. And this is important for translation efficiency, but it's also important for evading innate immunity. There are two nucleases of slightly unknown function, I would say, at this point. Berta will talk about the screens that, for these two. And then finally, there are two replication enzymes, the RNA helicase and the RNA-dependent RNA polymerase. I just want to make a point about the evolution of the virus that I think is quite important in this. So this little figure actually shows where the mutations were in the first major variant that came out, the so-called alpha variant or the Kent variant. What you can see here is that most of the changes are in this spike protein and in some of the other structural proteins. But in fact, in terms of the replicative part of the NSPs, there's virtually no mutations in it. And so actually these non-structural proteins are very highly conserved among coronaviruses. They probably can't tolerate a lot of mutations, and they're all mostly essential for the virus life cycle. And Raquel will tell you a little bit about her cell-based assays that looked at this. And so antivirals might not just be useful for SARS-CoV-2, but might actually be useful against other coronaviruses. And as Mark mentioned, we need to worry about the potential of vaccine-resistant variants coming out. So the project was to express and purify all of the proteins in E. coli and baculovirus, to characterize the enzymes and to develop assays that would be suitable for high throughput screening, and then to screen an in-house compound library that had more than 5,000 established pharmaceutical compounds. And then we characterize these from the screen and then test the best hits in viral growth assays. So I'm going to just quickly go through one screen, the screen that we did for the RNA-dependent RNA polymerase, just to give you a sort of a flavor for it. So we purified the proteins, we expressed various different combinations of these 3 subunits in insect cells and E. coli, finally settled on an active version. And this just shows an assay looking at RNA-dependent RNA polymerase. So we start out with this substrate that's a small primed oligonucleotide, and when it gets filled in, it makes this completely double-stranded molecule. And I think you can see here that with time, we get product accumulating. But this kind of an assay is really not very useful for screening thousands and thousands of chemicals. We needed to have an assay that would work in a high throughput format. And so to do this, and you'll see this kind of approach in all of the screens today, we used a Threatbased assay in which we have a fluorescent dye and right next to it a quencher. So this part of the molecule, the primer template, is pretty much the same as I've shown you already. And the idea is that as the RNA polymerase extends across, it can displace this oligonucleotide with this 5' flap. And when this oligo is displaced, that allows the Cy3 to fluoresce. And so we read that as a positive signal. And that just shows here some a titration of the enzyme, showing that the more enzyme we put in, the more activity we get. And it makes the point that we can do this as a kinetic assay, so we can calculate natural velocity, enzyme velocity from this. So we did this in a high throughput screen against all 5,000+ of these compounds. And this just shows the primary data here. You can see that most of the compounds up here and all the controls basically didn't inhibit the activity, but you can see a few of these dots down here, a few at the low concentration, a few more at the higher concentration, inhibiting the enzyme activity. And I'll just show you an example of one of these. It's a compound, it's actually a drug called Suramin. It's used to treat river blindness and other things. It turns out to have a fairly good IC50 of less than 1 micromolar. This also makes the point that the presence of detergent doesn't change the IC50, and this was an important lesson we learned in the screen, that a lot of the compounds aggregate and can give you non-specific inhibition, but real inhibitors aren't affected by the presence of detergent. And this just shows, using that gel-based assay, that Suramin is in fact a very good inhibitor of the RNA-dependent RNA polymerase. So I'm not going to go through this table in detail. If you want to look at it, it's in the group of papers that were published earlier this year. It's a list of all the inhibitors that we found. And just to make the point that these inhibitors range from sort of natural chemicals like dihydrotantionin, which you'll hear Kang Wei talk about, to some existing drugs like for example, Suramin, which is used to treat sleeping sickness, and trifluoperidol, which is an antipsychotic. So there's some characterized drugs, and there are some less characterized chemicals as well. And in almost each case, we identified more than one inhibitor for each enzyme, which will be useful in terms of toolbox. And in almost every case, these drugs also inhibited viral growth, which is what Raquel will talk about at the end. So just to summarize, this was our goal, was to screen these molecules. We think we have this set of compounds that will be useful tool compounds to study the virus. There are some potential interesting drugs for repurposing, lots of lead compounds for further development, and I think Jennifer will tell you a little bit about that. And then finally, this idea of identifying synergies between inhibitors, I think, is an interesting one. And one of the things that we did was to test each of the drugs in combination with the only really accepted antiviral remdesivir, and found that one set of them, the NSP14 methyltransferase inhibitors, did have some synergy with remdesivir. So that's really everything I wanted to say. This was a massive project. It involved 40 odd people, 35 of them from the Crick, a number of people from Dundee, most of them from the MRC PPU there, and it was a sort of a once in a lifetime thing that I don't think I'll ever forget. It was stressful, but it was also incredibly gratifying. So I think I'll stop there. Thanks.
Thank you very much, John, for that inspiring tour through enormous amount of effort. That's terrific. And just to remind people, the slide is up for questions for John. And for any of the speakers, if you could submit them in the question box as shown here, and just specify for whom the question is, and then we'll get to them at the end. So excellent, thank you. So let's move to our second speaker. Second speaker is Jennifer Milligan. Jennifer is originally from Fife in Scotland and joined John Diffley's group at the Crick Institute in 2018 to start her PhD. Her PhD project is mainly focused on studying mechanisms of chromatin replication using an in vitro system of purified proteins. That's what she does when she's not working on SARS-CoV-2. During the pandemic, however, her focus was switched, as John just described, to the enzymology of SARS-CoV-2 replication and discovering, validating, and optimizing inhibitors against it. Jennifer will tell us about a part of this project, identifying the 3CL protease inhibitors. So welcome, Jennifer, and we look forward to hearing your talk.
Thank you for the introduction, and thank you to the organisers for the opportunity to speak today. So I will be talking to you about one specific portion of this larger project, and that was the identification of inhibitors for the main protease, the 3CL protease from SARS-CoV-2. So just to keep a brief introduction, just to remind you, but the first 2/3 of the SARS-CoV-2 coronavirus responsible for COVID-19 encodes all of the 16 proteins responsible for viral proliferation, all of which are excised or cleaved from the polypeptide from which they are produced by two viral proteases. The papin-like protease, which Canway shall discuss, cleaves out NSP 1 through 3, so non-structural protein 1 through 3. And then the main protease, which is a chymotrypsin related protease encoded by NSP 5, cleaves out the remaining 4 NSP 4 to 16 at 11 conserved cut sites throughout the polypeptide. So towards identification of inhibitors, we first optimized a purification scheme that would arrive at pure, untagged, unmodified protease, 3CL Pro, quantity is good enough for screening. And we then wanted to assay activity of this protease. And one method that we did this was the development of a fusion protein designed from one of the non-structural proteins, which is not super important, but we used NSP9 here, attached to a flag His epitope, so something that was fairly chunky for size, cleavable by one of the consensus cut sites from the polypeptide, which I mentioned previously, the one in particular that we used being the junction between NSP4 and 5, which is the one predicted to have the highest affinity for 3CL Pro. So on the addition of 3CL Pro, this fusion protein is cleaved into two products which are differentiated by size. So they are suitable for analysis over an SDS page gel because we can see the two products. So on the first lane, we have no 3CL Pro and just substrate, and you can see that this does not cleave. And then in the presence of 3CL Pro and increasing over time, you can see an increase in product and decrease in substrate. However, this gel-based assay was not suitable for high throughput screening, so as John mentioned, we developed a Threat-based assay for protease cleavage. And in this instance, we have a fluorophore attached at one end of a short peptide with, again, 10 amino acids from the NSP4-5 cut site. And at the other end, we had a quencher. And when this peptide is not cleaved, the fluorophore and quencher remain in close proximity to each other, and so there's no fluorescence. And then on the addition of the 3CL protease, this peptide is cut, and the fluorophore and the quencher, the distance between the two is increased, and so there's fluorescence as there's cleavage. So we managed to scale this up for a high throughput capacity, and we could also measure the course of the reaction over time to get the slope of the reaction or the rate. And because we can look at thousands of compounds, we can then look for compounds that reduce the slope of the reaction or the rate of the reaction by a certain amount. And that's just what we did. So here I show a diagram displaying the normalized rate of reaction of all of the drug compounds, which was over about 5,000 at the highest drug concentration that we screened. And you can see here all of these dots underneath, which were compounds which seemed to reduce activity by at least 30%. So it was a bit more over the noise of the reaction. We got about 27 compounds from the initial primary screen, so 5,000 down to 27. And these were put forward for validation, which I'll just talk through briefly. So we removed the compounds that interfered with fluorescence, and what I mean by this is they artificially increased fluorescence, so they reached saturation of what we could measure quicker, and so they artificially had a low rate of reaction, so they were kind of false positives. After we went through that, we had about 13 compounds, which we could then do a secondary screen for. So this is in the Threatbased assay, but over a wide range of concentrations, and any real inhibitors would be reproducible in this, and we could calculate IC50 values. We got 12 compounds which we could do this for. Then we sought to validate these out with a threat-based assay. So we used gel-based assays, and it's as I just described, to make sure that out with a threat assay they could still inhibit, and we only have 5 from here, so 5,000 down to 5. And then we made sure they were specific for 3CL-Pro by testing against other proteases, the PL-Pro protease from SARS-CoV-2 and a pan protease, just like thrombin. And here I have shown the 4 final compounds here. 1 being a calpain inhibitor 1. And you can see that they all inhibit at all concentrations tested. So calpain inhibitor and also calpain inhibitor 2 and 11, which are inhibitors of the calpain proteases, were also identified around the same time to inhibit the 3-cell protease in vitro. So I'll just be talking about our other type of inhibitor that we identified. And this was all fluoromethylketone peptide-based inhibitors, which were all initially designed to inhibit caspases. So all of these FMK small peptide inhibitors had very, very good potency against 3CL-Pro, and in fact they were in the low micromolar range for IC50s. And they had a similar general structure of a Z cap at the end to help with cell permeability, a short peptide targeting sequence, which generally mimicked the protease active site for the proteases that they were designed for, and then a warhead molecule or a functional group, which would covalently bind to the active cysteine residue in these cysteine proteases. And in this instance, that would be a cysteine residue in here. So we wanted to see if we could increase the specificity for 3CL-Pro by changing or altering these inhibitors to improve them, given that they were designed against other proteases. So to do this, we looked at the sequence with highest affinity. Can you still see my screen? I have an internet problem.
You're a little bit fuzzy.
Yeah.
You can see your slide okay, but you're and here you're okay, but your face is a little bit low resolution.
That's fine. You don't need to see my face. So we looked at the sequence again for the NSP45 cut site, and we compared the inhibitors that we got in the screen, and we saw some similarities by property. So we then designed 3 custom inhibitors, but this time using the substrate sequence, which was from the cut site of 3CL-Pro with the highest affinity. One important point to make is that we couldn't, due to synthesis, have a glutamine here, so we have an aspartic acid here at the P1 position. And so from this, we designed 3 inhibitors with the same protective group, the same functional group, but differing by amino acids in the peptide targeting sequence. And we validated these and tested them as we did with the previous compounds. And we found that the shortest of all of them had the best inhibitor capacity, with an IC50 in the low nanomolar range. However, These drugs all have one major issue, and that's when they're digested by the liver, they release some pretty toxic byproducts. So they're not suitable in this format for drug use, but they do serve as an important tool for further development. And this is as their kind of Swiss Army knife type of design that is possible, i.e. you can change the cap to facilitate synthesis and cell permeability. You can, as we demonstrated, change the targeting peptide sequence, and also you can alter the warhead or the functional group and swap it for non-toxic alternatives. And we did do some work towards this, and we found an alternative to FMK, which is the difluorophenylmethyl group, or OPH, which we tested and we've listed in our publication. And it did have some inhibitory effect against 3CL-Pro in vitro, which would serve as a good starting point for further drug development. And so with that, I would just like to highlight again that this was just one part of a huge collaborative project, and there were many people involved, but I just wanted to acknowledge people specifically who helped with the protease, and this was Teresa, my co-first author, who did all of this with. Those from the peptide chemistry STP at the Crick, which were brilliant in helping us design and synthesize the custom peptides. Those from my own lab, who helped greatly with protein purification, assay design and optimization. High throughput screening, who helped us make it a high throughput screen and dispensing the plates. Raquel from the cell infection laboratory, who will talk about the cell-based assays, and those from Dundee who helped with protein purification. And lastly, I'd like to thank the organizers and the audience for listening. And I'm going to end it here. Thank you.
Thank you very much, Jennifer, for a very nice and very clear talk. It was impressive work. Good, excellent. And again, if you have questions for Jennifer, please submit them through the question box as shown on the screen right now, and I can direct them to Jennifer at the end of the session. So our next invited speaker is Dr. Kang Wei Tan. He's from Malaysia and obtained his PhD in Japan at the Nara Science and Technology Institute, where he was working on replication fork slowdown under checkpoint conditions in E. coli. He then came to the UK and worked at the Gurdon Institute at the University of Cambridge for a postdoc in Philip Zagerman's lab, where he was studying how phosphatases regulate replication initiation in budding yeast. And now in John Diffley's lab at the Crick, he's focusing on reconstitution, when he's not working on SARS-CoV-2, he's focusing on reconstitution of replication using in vitro approaches. and as you will hear, during the lockdown, he joined the team to help screen for inhibitors of SARS-CoV-2. So your slides look great, and where everything looks good, and look forward very much to hearing your talk. Thank you.
Yep. Hi everyone. My name is Kang Wei from the Diffley lab. So thanks for the introduction and the opportunity to present our work on this webinar. So I'm going to talk about the antivirus compound screening for the NSP3 peptide protease, which is one of the 7 papers that we published recently. So I just want to mention that this work is equally contributed between Chu Tianwei and me. So as a reminder, so PL-Pro is encoded within the NSP3, which is the largest NSP in the SARS-CoV-2. So to research on the PL-Pro, we use the truncated version that covers the UBL2 and the PL-Pro domain that also has been used in the previous study. So we express and we purify the PL-Pro from both the bacteria and the insect cell system in case the different post-translation modification is important. We then develop an in vitro protease assay, and we found that the protein purified from the bacteria are slightly more active, and therefore this was chosen for the subsequent experiment. And in this slide, I will explain in more detail on how the in vitro assay was set up. So as a reminder, PL-Pro is known to cleave the viral polypeptide to produce NSP123. and the rest of the NSP was produced by the main protease NSP5. So also, as Jennifer has explained, we utilize the FLAT technology to visualize the substrate cleavage activity via the PRPro recognized sequence. PRPro is known to cleave after the sequence LXGG, and once the cleavage occurs, the fluorescent signal is released, and this can be detected by the plate reader. So we observe the substrate cleavage reaction in the short peptide that contain the boundary sequence in between NSP2 to 3, but not NSP1 to 2. And this reaction rate can be significantly increased by using a bit longer substrate. And then having the in vitro assay developed, we then use it to screen for the PRPro inhibitor using the 5000 library from the high throughput. So we pre-incubate the incubator with the enzyme before the addition of the substrate. So this is a time course experiment where we could follow the reaction happen in the real time rather than just the endpoint result. And a representative of the normal reaction with the reaction containing a positive inhibitor are shown here. So we want to search for the good inhibitor that inhibit the enzyme with a low concentration. So in the screen, with just above 1 micromolar of each inhibitor, 8 inhibitors was isolated out of the 5,000, and they shows inhibition rate of at least 25%. We then repeat with the screen with the higher concentration of the drug, and we obtain more hit as expected. Next, we develop a second assay to validate those hits. So this is a gel-based assay instead of the fluorescent-based, where we construct the PRPro cleavage site on a much larger protein, and so that the cleavage product can be visualized on the SDS pitch gel. And indeed, the hit that we got from the screen also stopped the protease cleavage in the gel-based assay. We included some of the non-PRPro inhibitor as a positive control, and they stop PRPro activity at a various degree. We have to exclude some of the inhibitors, even though they inhibit the PRPro. And this is because they also appear in the other screens, and therefore shows the non-specificity. And finally, this assay efficiently ruled out the false positive hits from the screen, as they did not inhibit PRPro in this gel-based assay. And we find out that this is due to the autofluorescence issue. So after all, the candidates was reduced to just a few, which is easy to handle. Finally, we determined the IC50 of those inhibitors with a wide range of the concentration. So they are dihydrotensionol 1, RO082750, and the beta-lapatron. And they have IC50 less than 1 micromolar. So with dihydrotensionol 1 being our best hit, we also tested some of the tensionol derivatives. They are not present in the screen. So they also show IC50 just above 1 micromolar. We include a non-PRPro inhibitor, GRL0617, to see how does this look like under our experimental condition. And it has IC50 close to 2 micromolar. So from this screen, we found some good inhibitor that inhibit PRPro with the nanomole scale of the IC50. Next, we wonder whether those inhibitors are general protease inhibitor that could also inhibit 3CL-Pro, the main protease in the virus, which is also a cysteine protein, just like PRPro. So we tested those inhibitors on a similar gel-based assay, as Jennifer just explained, for the 3CL-Pro, with the cleavage product visualized on the SDS pitch gel. And novel 3CL-Pro inhibitor, ZVAD-FMK, that is discovered by the screen, effectively stopped the 3CL-Pro activity. However, none of the PRPro inhibitors stopped the 3CL-Pro even up to 10 micromolar. So from this, we concluded that those inhibitors are specific to the PRPro. So interestingly, PRPro cleavage site, LXGG, is not only found on the virus protein, but also present on the human protein, such as the ubiquitin and the ubiquitin-like protein, ISG50. And in this oversimplified diagram, when the virus attacks the hot cell, ubiquitin and the ISG50 are known to conjugate to the target protein, as part of the signaling pathway that finally trigger the immune response. So reversing of this process by the PL-Pro therefore has been suggested as the viral strategy to suppress the host immunity. So we did a test to see if PL-Pro is capable of cleaving the polyubiquitin. The answer is yes, as we observed cleavage of the trimer into the dimer and the monomer. So when PL-Pro inhibitors such as beta-lactam and the dihydrotensionol 1 was added into the reaction, this activity was largely inhibited. And the same observation was obtained on the ISG15 experiment. So in summary, the PL-Pro inhibitor that we discovered not only stop the viral replication, but also has the implication in restoring the host defense system. So dihydrotensionol 1 appear to be the best inhibitor in our screen, and it is a natural compound isolated from the Salvia mycorrhiza, as John just mentioned, which is a traditional Chinese medicine with a long history of usage. So together with the dihydrotensionol 1, cryptotenshinol and the tensionol 2A are also isolated from the same plant, and they share the similar chemical structure and show a comparable IC50 towards PL-Pro. So in fact, we also did the cell culture-based antiviral qualification assay for the SARS-CoV-2, and we found that only dihydrotensionol 1 is effective against the replication of SARS-CoV-2 with the EC50 of about 8 micromolar. So I think the next interesting question is to find out how dihydrotensionol 1 bind and inhibit the PL-Pro, and this has been proposed by a recent in silico study. So with this, I just want to say thanks to John, who initiated the whole project, and also he's also the supervisor in my lab. We always thanks to the high throughput screening for the 5,000 inhibitor, and for our project, we want to thank peptide chemistry for the substrate, and I also like to thank everyone else who joined the other 6 screens. So thank you.
Great, thank you very much, Kang Wei, for that very nice presentation. Really superb work. Good. And again, don't forget, if you have questions for Kang Wei, please submit them through the question box that we've described several times, and if you can see on the right of your screen right now. Good, and we'll pull them together at the end in our discussion section. So let's move on to the fourth talk. So the next invited speaker today is Dr. Berta Canal, who's a molecular biologist, did her PhD in the laboratory of Francesca Pozas in Barcelona, where she studied mechanisms that coordinate DNA replication and transcription during S phase to prevent genomic instability. She then moved to the Crick for her postdoc, where she obtained a Marie Curie fellowship to study how DNA replication is regulated in response to stress using biochemical approaches in the laboratory of John Diffley, and in her spare time has been working on SARS-CoV-2. So slides look good, everything looks perfect. So welcome, Berta, and look forward to hearing your talk.
Thank you, Mark, for the introduction, and hello, everybody. Among all the enzymatic activities of SARS-CoV-2, the virus encodes for 2 nucleases, an exonuclease and an endonuclease, and we managed during this big project to screen for inhibitors of both nucleases. And today I'm going to talk about the work we did to identify inhibitors of the exonuclease that is composed of NSP14 and NSP10 proteins. The virus encodes for the NSP10 cofactor and the NSP14 bifunctional enzyme that has exonuclease activity and methyltransferase activity. For the exonuclease activity, NSP14 has to combine with NSP10 and form a complex in order to have this exonuclease activity. It is not a common trait of RNA viruses to have exonuclease activities. It has been proposed that this exonuclease activity present in most coronaviruses serves the virus to evade the immune reactions from the hosts. And also interestingly, it has also been proposed that this exonuclease activity can serve as a proofreader of the virus, so that it would help correct the errors that the RNA polymerase might make during the genome duplication of the virus, and that this proofreading activity might have been involved in the enlargement of the genome that is characteristic of coronaviruses that have one of the largest genomes among the RNA viruses. Interestingly, our supposition at the beginning of this project was that if we were to find inhibitors for this exonuclease activity, we would be able, in one hand, to maybe cause the accumulation of some sort of RNAs in the cells that would be recognized by the immune system, and that would help with the reaction against the virus. And also, if we could interfere with this exonuclease activity with an inhibitor, we would also mess with this proofreading capability and maybe affect the growth of the virus. So the first thing that we wanted to do is to purify the protein, as it has been shown for the other projects. And the key that has been shown by others and that our preliminary data suggested was that it is not that easy to purify active NSP1410 proteins that have good exonuclease activity, and that the key for this exonuclease activity was the stability between NSP14 and NSP10 complexes. So we decided to do an alternative strategy and to fuse both proteins and purify a fusion protein. We developed an assay based on previous knowledge from SARS-CoV-1 exonuclease, in which a label template of double-stranded RNA would be cleaved by the exonuclease, and the cleaved products could be then visualized in denaturing polyacrylamide gels. So using our purified fusion protein, we performed this assay where we could visualize the substrate and the cleaved products in such gels. And interestingly, what we saw was that our fusion protein was very active, a lot more active than what previous purified proteins, exonuclease proteins, had been shown in other projects. So we decided to use this fusion protein to perform our screen. The other key aspect of the project was to develop an assay, though, that instead of being run in a gel, could be assayed in solution, so that it would allow us to monitor exonuclease activity over time and without the need of a gel. As it has been explained by John at the beginning, what we did is to place a quencher close to our fluorophore, and that upon cleavage by the exonuclease of this double-stranded RNA substrate, the quencher would be released and we would be able to monitor fluorescence appearance. Using this substrate, we were able to monitor the activity of NSP1410 exonuclease over time to calculate slopes of the reaction, and with this estimate enzymatic activity. When we perform these reactions in 384 well plates containing the different compounds of the chemical library, we then would be able to measure slopes in the presence of each of the compounds and look for compounds that reduced the slope of the reaction, which could mean that they were inhibiting our exonuclease. We performed the screen and analyzed the slope in the presence of each of the compounds at 2 concentrations of the compounds again, and identified some compounds that were reducing the activity of, or putatively reducing the activity of our assay, and we identified some more compounds at higher concentration of the compounds. We curated all the results in order to produce a list of primary hits from the screen, and decided to validate these hits using the gel-based assays, which we considered was a more direct way of monitoring exonuclease activity. Just to remind you, in the gel-based assay, we can visualize the substrate and the cleaved products when the enzyme is present. When we perform these reactions in the presence of the selected hits from the screen, what we saw is that most of the hits did not alter the activity of the NSP1410 exonuclease in this assay. However, patchoulin and orin tricarboxylic acids, 2 of the compounds, did inhibit the exonuclease activity of NSP1410 in this assay. So to further characterize the exonuclease activity in the presence of patulin, what we did is to assess NSP1410 exonuclease activity in the presence of increasing concentrations of patulin, which allow us to monitor, to quantify or to calculate, half maximal inhibitory concentration of patulin that was of 1.8 micromolar. Patulin is a cyclic lactone that is produced by molds, especially in rotten fruits such as apples, and it has been shown to be a mycotoxic and to have antibiotic effects, and also, unfortunately, to be a highly cytotoxic molecule. So because of its toxicity, we don't anticipate that patulin might be a very good inhibitor to treat COVID-19 per se, but it could be very interesting to study derivatives of patulin or structural analogs that might help understand how the enzyme can be inhibited, and also maybe to use patulin in vitro assays in order to further explore the biochemistry and the function of the SARS-CoV-2 virus in research projects. Regarding the ATA inhibitor, we also calculated the IC50, which we found to be a little bit higher than the one of patulin, but still in the low micromolar range. ATA is a known nuclease inhibitor, so it has been shown to inhibit other nucleases such as RNase A and benzonase. However, when we compared the inhibitory role of ATA over RNase A and benzonase and compared it to the one of NSP1410 exonuclease, we found that NSP1410 was inhibited at much lower concentrations of ATA. So there was a window of opportunity there compared to these other nucleases. Interestingly, ATA has been shown to be an antiviral compound against other viruses such as SARS-CoV-1 and the Zika virus. And this is intriguing because Zika virus does not have an exonuclease like SARS-CoV-1 or SARS-CoV-2. So overall, all this knowledge about ATA suggests that this compound might have effects on different compounds. So it could be inhibiting, for example, NSP1410 exonuclease, but it might have roles in inhibiting other important factors, for example, in the host cells, that could help in an antiviral role. So we believe it's an interesting drug to further explore, especially considering that it's very low toxicity. To finish with this part, I want to acknowledge and to thank especially Alison McClure and Joseph Curran that are co-leads in this project with me, the whole lab, of course, and all the people involved in the project, people in the Crick and people in the University of Dundee, all the facilities in the Crick and the funders, and all of you for being here and for your questions.
Thank you very much, Berta, for really very nice description of that really cool work. Good, thank you. And now, again, if you have questions for Berta, please do submit them using the box that you can again see on the screen, and we'll come to them after the last talk. So let's move on to the final talk of the webinar before we get to questions. Which is our last speaker is Dr. Raquel Hilfer, who will tell us a bit about the virological aspects of this and the effects of the compounds on SARS-CoV-2 replication. So Raquel got her PhD in molecular virology from Queen's University Belfast for work on replicative enzymes of SARS-CoV and related corona and nidoviruses. And she then worked on picornaviruses and influenza viruses to get more virological expertise at Utrecht University, and then was then at the University of Cambridge, and is now at the Crick in London. And her work includes assay development for screening antivirals, identification of antivirals that target replicative enzymes as well as host factors. And she's currently interested in the host factors of influenza and coronaviruses, as you'll hear a bit about in just a couple of minutes, and in particular membrane trafficking pathways with a focus on autophagy. So your slides look good, and I can see you clearly, Raquel, and I look forward very much to hearing about your work. Thank you.
Okay, well, thank you very much for the kind introduction, and thanks for organizing this very interesting webinar. So I'm here to now talk to you about a high content microscopy screen for inhibitors of SARS-CoV-2. As you've heard from John and his colleagues, they'd come up with really this amazing array of different inhibitors for these 7 different replicative enzymes, and thanks to Berta for this very, very lovely depiction that you 3 have seen throughout. But the question was now really, would these inhibitors that they'd identified in vitro also work in a virus infection, at least in cell culture, this being the first hurdle for then any antiviral. So if we're looking at developing a high content screening assay for inhibitors of SARS-CoV-2, what are we looking for? Well, as minimal handling and high containment as possible is helpful not only for safety reasons, but also because due to all the safety requirements, it is rather slow, the work that you do there. For a sort of similar reason, potential for a maximum optimization, and at the same time be able to prize apart whether it's actually antiviral activity we're looking at or cytotoxicity that this compound causes. And sort of just briefly as to what kind of methods are commonly being used for those kind of assays. Not necessarily all of them. The first being for a virus that, like SARS, causes a so-called cytopathic effect, so kills the cells. As you can see here, the cell culture infectious doses, CCID50, which there's little chance of automation. And I've spent more time than I would care to remember in reading these out under a microscope, while the sample is largely remains infectious. It's not exactly fast, and you would still require a separate readout for toxicity because that's your endpoint for the virus. Then, well, a currently standard used assay, generally plug assay to quantify, can also be modified, shown here, the plugs, the white points. Some automation possible, but limited. Really has a high amount of handling, and it would take 3 to 4 days for visible plugs for this virus. Now, qPCR, you're probably all familiar with. Very early on, it was possible to to have an assay to quantify that. It's arguably the most direct readout of replication. Because you are looking at RNA, the automation is possible but limited, but you have a lot of processing steps, and for this level it really becomes very expensive because the reagents are very expensive. Now, one that we were particularly interested in is an antibody-based detection of viral products with potentially a fluorescence readout, though you could use other methods in this case. However, the snag being, well, antibodies against a novel virus. I mean, it takes time to produce these, and taken from sort of one of the early descriptions, it certainly is a novel virus. It's distant enough from SARS-1. However, overall, there is still a good enough similarity between the on the protein level. So we reasoned that we could very likely use SARS-CoV-1 antibodies that would then cross-react the SARS-CoV-2. And as you can see here, that panned out. We tested a variety of either non-structural or against structural proteins. Now, one sort of the odd one out. There are antibodies that were generated against a plant virus that will detect double-stranded RNA. So this is something that so far most RNA viruses do appear to make a surprising amount of. So this could be used. We were particularly pleased to see that this antibody against the nucleocapsid CR3009 worked for the following reasons. So this was an antibody from a panel that was described and isolated against SARS-CoV-2, and our colleague at UCL, Laura McCoy, had the expertise and the foresight to clone these antibodies. And then we're very fortunate here at the Crick to have we're calling the service technology platforms, and Svenkia from the structural biology STP could rapidly produce large amounts of this antibody that could be directly labeled, so it became cost efficient. And this is the general setup of this assay that we came up with. So you would have your cell of choice, add a dilution series of drugs, then add the virus to this, and this part, growing the virus, and this would still be done in the high containment facility. And then fix the sample, and at this point, the virus would be inactivated, be safe to take out of the facility, and can then stain for the virus with this antibody and for the nuclei as a simple readout of cell number and therefore toxicity, and analyze this by microscopy. And again, luckily, Mary Wu from the high throughput screening STP has access to all kinds of very fancy robots and equipment to automate most of these parts, including the automated high content microscopy. So, and this is what this looks like. This is screening at 5x. You can see the infected cells, several foci. At 20x, you would find, well, you can start telling the individual cells. However, we found that this was something that Mary worked out, at 5x magnification, this was sufficient to really analyze this just simply by looking at the green positive area and quantifying that, which made this very fast for screening purposes. And as proof of principle, now you've heard this mentioned before, at that point, pretty much the only one available, remdesivir, the drug that works on the polymerase by being incorporated. And as you can see, we got EC50s in the low micromolar, depending on the experiment, which is in good agreement with the high nano to low micromolar range that has been described in the literature. Now going back to just some examples of the ones that we tested. So first of all, the one Berta introduced, the inhibitors of NSP14 exoribonuclease. So patulin as well as ATA were found to inhibit the virus in cell culture. John, as he mentioned, was particularly interested in whether there would be some synergy at that point with remdesivir. Most reason to think that that might be the case for the exoribonuclease. However, in this case, we did not find any synergy. So the inhibitors of the NSP5 main protease, the calpain inhibitor, had been previously shown to inhibit the SARS-1 MPRO, but no data on whether this would work in cell culture. And as you can see, it works with really low EC50. Then this elegant series of main protease inhibitors with the modification. And again, while the original one sort of very low activity, there was some improvement of this and certainly further room for further improvement. However, also no synergistic effects with remdesivir. Now for NSP3, the papain-like protease, dihydrocantinone also worked out to work very well in cell culture. However, as you can see, cryptotanghinone and tanghinone, so the similar molecules that had been described previously to work in vitro and which were confirmed to work in vitro, really no effect in our assay as far as we can see, showing that's really important to follow this up. And then also the other inhibitors showing sort of reasonable EC50 values, so good starting point for further development. With GRL being described, having been described previously. Again, no synergistic effect with remdesivir was found here. And well, this is one of the reasons why I bring up an additional screen. So as John mentioned, a lot of these viruses, well, basically they provide their own capping enzymes, one of them being NSP14. And the series of inhibitors that were found to inhibit and work quite well in cell culture. Here, the value from the previous without remdesivir shown in red. You can see that there is some improvement if we include remdesivir in this. So just, as I said, a brief overview, and if you're interested, all of the other ones are published. And well, as I said, it was a large consortium, so thank to everybody. It's really amazing work and very exciting. But just for sort of this particular, for the screen, I want to particularly point out Agustina Bertolin, who basically coordinated between the in vitro work and the in vivo work. And I've mentioned Mary Svend, the WHO Influenza Center, which helped with their expertise on working in a CL3 environment. And as I said, Laura for providing this antibody, which can be obtained here. And thank you very much for your interest, and I'm happy to answer any questions.
Thank you very much, Raquel, for that comprehensive view of the key findings of what these drugs and potential drugs can do. That's terrific, excellent, good. So that's the last of our scheduled talks. So now we can have some questions. So we welcome questions for the speakers. We have a few of them, and as mentioned, if you have questions for the speakers, please pop them in the box that you can see on the screen. Okay, good. So thank you. So all the speakers are now back, and I will share some questions. So just to start, there's a Darja Demoneva had a general question, which was, given the conservation of the non-structural proteins, could antigens in them, presumably processed, be used as potential vaccine targets in these cases? Has that been done? So, John.
That's a good point. I think the real problem with that as a concept is that they're never outside the cell. So they're intracellular. They're expressed only in cells. So I think antibodies obviously work best against the structural coat proteins.
But if there's enough protein, what I was thinking when I was looking at the question was that if they're processed and presented by MHC and so forth, could there be effectively a neoantigen mode for them to be exposed?
Yeah, I think it's not. It's probably a legitimate thing. I think it's also worth saying that there are lots of other proteins on the coat of the virus that haven't even been exploited yet, which I think would be worth thinking about, because unfortunately the spike protein is the one that evolves the fastest.
Sure, indeed. Good. And
yeah, and sorry, maybe
ahead.
just to come on to that. So, I mean, they've certainly, you can find that if you look in the blood, people will produce antibodies against other structural proteins, also against some of the non-structural proteins. But as John pointed out, it becomes a question of sort of as a vaccine, how, I mean, the people are working on this, but the spike certainly is sort of the most prominent target.
And the average cell, I think, will have very few of the NSP antigens presented on its surface anyway. But good. So thank you very much. So another quick question from Asha Tiwari, which is the question for the NSP14-10 fusion. The question about whether the exonuclease activity becomes more active does the fusion enhance the exonuclease activity of NSP14 directly?
Well, yeah, I mean, I guess I'll just chip in something, which is that there's two issues. One is that the complex between NSP14 and NSP10 isn't very stable. So there's some element of the fusion simply helping the complex formation. I don't know if Berta wants to say something else. Whether there's really a direct effect on the nuclease activity, I don't know.
Yeah, well, I agree. I don't know either. One thing that I wanted to say regarding this instability of the NSP14 and 10 complex when they are alone is that it's supposed to, in cells, to form a complex with the other proteins, NSP12, NSP7, 8, etc., to make like a replisome complex. So the idea of fusing them together is to keep them close, because we believe, or there is the suspicion that this instability of the complex is not a normal thing either. So it would be a non-physiological thing anyway. So we thought that we might be losing some inhibitors by fusing them together, maybe something that would impair NSP10 to be close to NSP14, but still we might be winning some other things because having unstable complex would be problematic.
Yeah, I think it's interesting too that NSP10 is a small protein, but it's also a cofactor for the other cap methyltransferase, NSP16. So it may be, as Berta said, that there is some higher order complex that gets formed that helps stabilize everything. I think it's really, I think one of the things that's amazed me coming into this whole thing is how little we actually really know about the replication of coronaviruses and how much there is to learn.
Right.
One other thing that I didn't explain is that what we did once we had the fusion protein is to assess methyltransferase activity, because in theory, having or not NSP10 around, NSP14 should be able to have methyltransferase activity anyway. We did the test in this regard using SAS and TIF that were involved in the methyltransferase screening managed to assess methyltransferase activity of the fusion, and they saw that there was a normal, comparable methyltransferase activity of the fusion.
Okay.
So this was not affected.
Sure. Okay. Great. Thank you. And next, I have a question from Mark Duffy. A question for Jennifer, which is what are the next steps for developing the warheads, the new warheads, the OPH, I think, warheads that you had identified, and then what are the prospects with that?
So the development of the OPH peptide inhibitors would be an exciting project that's not really in the scope of what we're doing anymore. It would involve some kind of novel chemistry, as the addition of the OPH group to the peptide linker is not simple, especially if you wanted to make the additional improvement of having the glutamine in the P1 position. So that in itself would be a very interesting chemistry endeavor, as well as creating what could be a very useful and novel drug. And I would implore anybody who wants to have a go at it to try. But that would really, in my opinion, be a really cool next step. You could try the FMK warhead with the Q in P1 to see if there's an improvement, and then try for a non-toxic alternative. There are others, not just OPH. That just happened to be the one that we could source commercially in a inhibitor that we could test. Yeah.
Thank you. And good
Maybe
plug.
I add something that, you know,
You do.
we're kind of out of this business now a little
Yeah.
bit, but all of the reagents are available to anybody who wants to take any of this on. All the constructs are available at AdGene, and we're available for consultation on any of this if anybody's interested.
And it kind of related to that, another question actually from Mark Duffy for Raquel, which is, are there plans to look at synergistic effects with the latest promising antiviral molnupiravir? Or should Mark do that?
So, I mean, it's certainly possible. We've not directly planned doing these, but yeah, I think especially given that we saw very little synergy with sort of any, like especially the exonuclease compounds, I'd be very curious to see whether a different ribonuclease, a different polymerase inhibitor would make any difference there. And also the mechanism then behind that would be very, very interesting to unravel.
Thank you. And actually, also for you, Raquel and John, Blair Strang was asking whether you'd excuse me, attempted to generate drug-resistant virus mutants. And I think, just summarizing the question, I think that the idea is that if you put them on, if you put the virus under selective pressure, will you actually, even though they're well-conserved proteins, will you get mutations in the non-structural proteins?
That's a great question, and I guess that's a sort of a generation 2 kind of experiment. I guess before you go anywhere, you'd want to know how easy it is to get revertants or resistant mutants, but we haven't done that. Good thing.
Good, and then I think the final question I have here on my list, I had a couple of quick ones I want to ask. Two was for Bertha from Vijada Das, was, had you tested, or has anyone tested patulin on the cellular counterpart enzymes?
I'm not sure I understand the question.
But I think just in terms, I think it's really a matter of, I think the question, my sense is that's all I have. My sense is that the question may be about selectivity versus related cellular enzymes.
So I don't know. One thing we tested is how patulin might inhibit other nucleases apart from the NSP1410. So we tested.
I think that's the question.
Okay, so yeah, we tested whether patulin was a general nuclease inhibitor, and it might also inhibit RNase A or benzonase. And what we found is that not up to 50 micromolar. We didn't test further from that, but we didn't observe any inhibition of these other nucleases. And what we saw in the cell-based assays that Raquel and Mary did was that there was some toxicity as it had been reported. So which are the targets there, I don't know.
Okay, great. And another question for also for Kangwei and other protease people. Thinking about synergy that you'd seen with Resdemivir, is there any? Do you think there might be selective synergy for subclasses of targeting effects? Effectively, I guess what I mean is that, do you think could it be that targeting multiple proteases, for example, would give you more synergy? And in a sense, and I guess related to that, is there a sense that the proteases do actually cooperate in any way, or are they entirely independent?
Yeah, it's a good question, because I think in both proteases, we don't see the synergy effect between the remdesivir and the protease. I mean, in biology, you may think that they work on the same pathway. That's why we don't see any additional effect of this drug.
But what I was more thinking is, if you hit multiple proteases, would you be more inclined to see synergy? That was really my question.
Yeah, I think absolutely. I mean, that was one of the purpose that we want to find a specific drug for each enzyme. And then if we combine all of them, and then we are able to kill every pathway in the replication of this virus. And then this is definitely a very good idea that we also want to do.
Okay.
Yeah, can I actually add a couple of things to that? One is that we had remdesivir. That was the only drug we had at the beginning, so that's why we tested everything in combination with that. But I think there's definitely a piece of work going ahead where we test all of the drugs that we've gotten in combination with each other.
Yeah.
I think that's a really interesting project. The second thing that I wanted to say is that we did all of these inhibition experiments in Vero cells, and Raquel knows more about this than I do, but I want to say something. But they have a very poor innate immune response. And one of the interesting things about all of these inhibitors is that they might be predicted to generate intermediate molecules that might trigger innate immunity, like uncapped messages, partially replicated molecules. And I think that there's a whole body of work that would be very interesting to do in perhaps cells that have a slightly better or stronger interferon response or general innate immune response. I don't know if you want to say anything about that, right? But I think that would be an interesting way to go.
Any other comments?
I have a comment on that. I did not explain anything about the screen of NSP15, which is the endonuclease. This is the only one of the enzymes that we screened that we found a putative hit in vitro, but that did not show any activity in the cell-based assays. And one possibility could be that because NSP15 has this major role in immune evasion, it could be that we did not observe any inhibition in the cell-based assays because video cells are not responding to that. But there is a window of opportunity for that, I think.
Okay. Can I just ask one final question, which is that there's been a plethora of in silico screens for potential inhibitors of these enzymes, and I'm an experimentalist, and that's what I need to say about that. But I was surprised back in the HIV protease days that Pat Kuntz and others managed to get very good protease inhibitors of HIV protease through doc and so forth. And I was wondering if any of you have actually looked through the in silico screens for overlap with what you saw with the compound classes that you looked at, or are they completely disparate? Is there any connection?
I think Kang Wei mentioned that dihydrotantionone came out of an in silico screen. Is that right, Kang Wei?
Yep. I think, yeah.
Tantionone's general. So I think there is some, has some success rate with that. Having said that, we've also been contacted by lots of people who've done in silico screens, and
I imagine
we've tested lots of their compounds, and we've not yet found anything that's inhibited. So I think it's, I think it's a good way to go. But I think as you said, the experiments are important too.
The proof is in the pudding. I always think that's why I'm an experimentalist. But
anyway. We did a sort of comparison with in silico screens that had been published for NSP15, because it was the screen that had less hits. So what we did is to compare what was known, what was proposed. And we found that some of the predicted inhibitors ranked quite high in our screen. So some of them seem to be also high in our screen. And these inhibitors were also RNase A inhibitors. So they seem to be general nuclease inhibitors.
Okay, good. Okay.
This is in the paper.
Right, great. Thank you. And I'm being asked to wrap up since we're pretty much out of time. So I'd like to thank you all very much indeed for fabulous talks of a summary of what has been extraordinary work. And thank you for publishing it in the Biochemical Journal also. And also thank the audience for coming and for their questions. And as you'll see on the slide at the moment, feel free, please, to continue the conversation online with the different Twitter handles that you can see on here. So, I'd like to just once again to thank everybody for attending, and again to thank the speakers very much for doing really a terrific job. I think it's been a really exciting webinar. So, thank you. Okay.
Thanks.
And if you have enjoyed today's session and then and would like to know more about the topic, you should definitely take a look at the collection on the website. You can see it right here, and you can in the journal from a short while ago, and you can our group of 7 papers with perspective and so on and so forth. And going forward, we welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So if you have an idea for a webinar in 2022, please submit a proposal, and you can see online at the moment how to do that. I'd also like to mention that if you have missed any of the 30-odd webinars that the Society has run as part of, and Portland Press has run as part of this series, or would like to watch them again, you can visit the website or the YouTube channel. You can see those on the right, those links, so you can actually go back and digest this one too, and at www.biochemistry.org/webinars. And then I'd like you to just advertise the next one. So please join us next time for a Biochemistry Focus webinar series entitled sPRNA-seq Challenges and Opportunities, which will be held on Thursday, the 28th of August. Sorry, Thursday, the 28th of October at 2 p.m. British summertime. Is that right? I used to be in Britain, but I can't remember what BST stands for. But BST. And doing this session is chaired by Professor Helen Whedon at the University of Glasgow, and the speakers will introduce the most popular microfluidics approaches, 10x, and also outline the main steps of standard sCRNA-seq analysis. And you can register now for this free webinar. And then just as a final point, I just want to make a comment about the Biochemical Society that, particularly during these times when, as you can see from the speakers, we're stuck in our various offices and homes and Zooming from places, the connections we get from something like the Biochemical Society are really important, whether you're a student, a postdoc, faculty, or none of the above. And so join, and you can find out more at biochemistry.org/membership, and you can join the big international group of biochemists represented by the Biochemical Society. So I think that's what I wanted to say, and I want to finish again by thanking everybody for attending, and in particular for thanking John for leading this sterling effort, which has been quite inspiring, I have to say. It was really a great pleasure to handle the papers for the Biochemical Journal with Dario Alessi, and to see them come out and see the journal get a chance to publish something that's really very important for all of us. And to Raquel and Berta and Kangwei and Jennifer for giving all terrific talks after John's today. So I think that's it. And thanks again, and keep up the good work, everybody.